Got it. Okie dokie. So this afternoon, we're going to go through the protocol one chunk at a time. And there's a lot of detailed information here in this thick packet, which is your volunteer handbook. The first 2 pages are sort of like some background information and just general guidelines. And then starting on page 3, we go into a whole lot of detail that you can follow along. We've tried to make the presentation really follow this packet so that you can take notes on anything that I say that's not written down that you might need to remember. Where I want to start with all of our protocol stuff is that it's sort of a big picture of where we've come from and how we've come up with these ideas. You know, to a certain extent, we come off as armchair scientists. We sit around a conference table and we say, what would be a really good way to test for green crabs? But at the same time, we know that these activities have to occur in the field. And Sean and I in particular have a lot of experience translating what's on paper into the field and dealing with conditions like running off the mud flat because the tide's chasing you back, and dealing with just really cold nighttime tides. We're not sending you out on nighttime tides. We don't want you out on the mud flat in the dark. So we've tried to apply a little bit of our own filter to these methods to keep you from having to be in uncomfortable situations. But we do want the protocol to be scientifically rigorous. The goal here is to get information that we can use to inform management changes and policy changes. If we can't prove that green crabs are causing these community changes, no one's going to dedicate money to controlling them and putting efforts into eradication. So the data that we collect will have a direct effect on how resources are allocated towards management of green crabs. So we want it to be reliable, high-quality data. We want to be able to make comparisons over space and time, so we want to make sure our big group of volunteers is doing the same thing as much as possible. We want to make sure it's a very sensitive test for detecting green crabs, so if we're not finding green crabs, we want to be confident it's because they're not there, or they're there just below detectable limits. But we also want this to be doable and engaging for volunteers. So we've updated the protocol from last year based on extensive feedback that we solicited. Last year was our pilot year. We put a lot of effort into focus grouping and getting feedback, both anonymous and in sort of group situations, about what things were really hard. We've brainstormed about how to make them better, hopefully arrived at something that is more comfortable this year. Not that no one dropped out last year because they thought it was horrible. I'll tell you that. I'm not trying to scare you away. We have had very good retention for the volunteers who participated last year. But we also welcome your feedback about things that are sticking points, tools that we can give you, or information we can give you that will make it easier for you to do your job out there in the field. And we won't necessarily be changing the protocol every year because that makes it very hard to compare changes over time. So there might be small changes from time to time, and we'll do our best to inform you, but there's likely not to be dramatic changes across years in the future. So the other sort of housekeeping thing that I want to do is just cover a few safety basics. It sounds like a lot of you have spent time in the mud and on shorelines doing surveys, so a lot of these will be second nature to you, but just to make sure that we're all on the same page. One really important thing about working in any field conditions, but especially in muddy areas is always having somebody with you. So we put you in teams because the work is easier, but also for safety. Even if your team member were just sitting on the shore, drinking a beer, watching you, we want somebody on the site with you just for safety. You might pick a better field helper, somebody who can actually at least write something down, but at the very least, somebody who can keep an eye on you and be somebody who can help you if you need it.
Does that person have to be trained?
So, no. So we want everybody who's participating in the actual data collection and monitoring to be trained through us. We know that there might be some people who maybe couldn't come today who will be getting training from us in different ways. We'll provide them the material, some online resources, And we also come out and do site visits, and that's another way we can communicate the information. But at all sites, our goal is to have the core members be trained by us in this area, and that's actually something that's written into our permit, that the people who are allowed to possess the permit have to participate in training. So it is a little bit codified that the training is a critical part of this. Another safety first item is just knowing your mud and your tides. You'll get to know your sites really well, and getting familiar with the whimsical nature of mud and knowing where the soft, sticky spots are going to be at your site, and how that changes, how the density of the mud changes as the tide changes, is really important to avoiding spots that could be hazardous. We don't want you to go anywhere that you don't think is safe to go, because we don't want to make you uncomfortable. So one of the tips I always give is obviously avoid the soft spots, but how can you tell by looking what soft spots are? So one key about telling what spots are soft is that they often have a little bit of standing water, even if the tide's all the way out. So that's more likely to be like quicksand, which that sounds bad. So avoid that. Walking on your toes, this is the part of this presentation where I get to look, keep moving, okay? Walking on your toes and moving continuously are ways to avoid getting stuck. So it's tempting to want to use your feet like snowshoes and present the greatest possible surface area, but what you're doing is actually creating a very large suction surface for the mud. So you then have to pull your foot back up, and if you have this big sort of perpendicular surface area, you've created a very large surface area for a vacuum to form. So walking on your toes, sort of like I think of an egret, they're very good at it, and continuously moving are ways, if you find yourself in a sticky spot, that's what you want to do. Just keep moving. The longer you stand still, you're just going to keep sinking, and if you don't get moving quickly, you're going to sink again. Obviously, if you get in a sticky spot, avoid it in the future. Get out of it and stay out of it. Don't go back there to try to put traps there again. I've put this tip up here. You can have your gear with you that can be used as leverage to help get you unstuck. And particularly, we give you these sorting bins. And this is something that you can use as sort of an external foot to lean on it as you're pulling yourself out. And you can use it as a granny walker to get yourself across. It's very effective. It's less dignified than, other ways of getting around, but more dignified than ending up stuck in the mud. So it's all about doing it with style. So dressing appropriately is key to being comfortable while you're doing this. No one wants to be freezing and wet while they're doing this. And the most important part of being safe and dressed appropriately in the mud is your footwear. I'm guessing I don't have to convince anybody here that they should not go barefoot into mud flats. Okay, good. So mud flats, it's very dangerous to go barefoot because there's often broken bits of shell in there. So while you might not lose your boots, you could easily slice open your foot in a muddy situation, which is then ripe for infection. I prefer narrow-ankled boots. I actually wear hip waders almost all of the time, even if I'm not going to places that are very deep. And in particular, I can kneel when I'm wearing hip waders, and so that makes it more comfortable. Chest waders, even better. You can just sit right on your bum and record data and not worry about getting wet. Narrow ankle boots make it easier to pull your foot up if you have these big galoshes. Nice and easy to step into and out of, but also easy to get left behind in the mud. So that's my 2 cents. Of course, sunscreen and potentially simultaneously rain gear, whatever keeps you warm and dry this time of year, super unpredictable. Cleaning your gear is really important. I put up this logo here. We are, of course, an invasive species awareness raising group, and so we want to be mindful of the role we play as volunteers and as monitors moving between habitats. So your gear will mostly stay with your one site. You won't be responsible for moving species around with your gear necessarily. But remember, if you go to another beach with your boots, maybe doing sampling somewhere else, Padilla Bay and some local areas are places where Spartina, the cordgrass, invasive cordgrass control is. So there could potentially be seeds in the mud that's dried in your boot treads. So it's really important to clean your boots on the beach when you get out as much as possible, and then at home, further clean them, especially if you know you're coming from an area that does have Spartina, using things like bleach and at least fresh water and making sure you get the tiny seeds and stuff out. Carrying a copy of your permit, not so much about safety as it is about just being in compliance. We give you a permit, it has to be in your possession for it to be in effect and for you to have a green crab. This is something that gets renewed every year, so every year we'll give you a new one. And just generally being good stewards of sites. Many of these are popular places that a lot of people like to enjoy, and so obviously not leaving trash. And being mindful of where private property boundaries are, certainly for your own safety, but also so that we maintain good relationships with property owners. We have been working to secure permission to work at a lot of these sites because some of them are private property or tribal land, et cetera. So communicating with there's a site near here that they've asked to be notified before volunteers come to the site so they don't think that there's weirdos mucking around in their lagoon. And so being in good contact and maintaining good relationships is an important part of just greasing the wheels, greasing the skids is I think actually the expression, for good future relationship with the site owners. Okay, now the nitty-gritty. As I said, you can follow along starting on page 3 in your volunteer handbook. But to give you the 30,000-foot view of what we do, I'm going to go over this in an outline and then I'll dive down into the details. So we are trying to capture the live crabs and fishes that live in the communities to get an idea of what's living there. We use 2 types of traps. These really common minnow traps that you might have seen, you might have even used them before. These have a small mesh so they capture the small organisms, including the young green crabs. We use a larger Fukui fish trap, which is a folding trap. It's made to be light with this very light plastic mesh and portable with a nice handle. So it's easy to carry relatively easy to carry a few of these out onto the mud flat. This is a type of trap that's used in a lot of green crab sampling. It's used frequently in scientific sampling because it rarely has to withstand really rigorous conditions, and it's light and portable, so you can just muck on out there and schlep it a little bit. Mesh is larger, so you get, and I should show you, the mesh is larger so that you get larger individual crabs going in there. They enter through the sides here. And you'll notice when you get to playing with these traps that we have zip-tied the opening to be a little less wide. And that's because we have tasty bait in there, and there are certain terrestrial predators that we're not interested in capturing, like cats and raccoons. So there's a couple things that we've done to try to minimize the possibility that we could get them in our traps. And we'll talk about that as well. So each time, each monitoring site will set 3 Fukui and 3 minnow traps each time they go. Monthly, April through September, so April will be our first month of monitoring. The traps are baited, and we set them so that we capture an overnight high tide. This is, again, when crabs are most actively foraging and attracted to bait and looking for food and willing to take risks to get it. The next day we return and we count and identify all of the organisms in there. So you guys are clearly all experts in everything we're going to catch from Sean's excellent talk. And so we're going to count everything in there. A subset of the crabs, we're going to take size measurements on them. And the reason we're doing that is that will give us information about the age of the crab population. Do we have a bunch of really tiny crabs, indicating that we've just had a recruitment event, a reproduction event, or are they all fairly old and there's no reproduction going on? This tells us about the age structure of the population. Everything gets released. So one key component of this, and it says it on the permit this year, they want everything to be released in the best condition possible. They want us to try as hard as we can not to kill things that we catch, unless, of course, it's a green crab, in which case we'll go through the specific steps of how to report them, but they don't go back out into the environment. We keep them because we have that permit, and we can do that. Okay, so that's the broad overview. One of the key first steps, the key first step, is picking a time to set your traps, a day at a time. And the reason that this is a little tricky, we're not assigning dates and times because the tides will vary at your sites. So these are traps that we're setting from shore. We're not, there are, not like if you went out recreational crabbing and you'd throw a pot deep off a pier or off a boat. These are shore crabs that live at high elevations, so we're setting the traps near where the crabs would be from land. So it depends. We have to look at the tide calendar to determine when the tide is low enough that we can walk out to set our traps, and how often the traps will be fishing. So I'll walk you through this. This is an idealized 2 days with night is the shaded in gray in daytime. And of course, because it's the Northwest, we have higher low tides and lower low tides. And when we set the traps, our rule of thumb has been to try to get them down to about +1 feet above mean low or low water. And that will vary slightly depending on if you're in a lagoon or not. I want to point out that a lot of this stuff, the specifics of timing and everything, we will go through with you at your site on that first day of sampling. We come out with you, we look at the site, we say where is the best spot to put the traps, and what are the timing considerations that are relevant at this site. So I'm going to give you sort of an ideal scenario, and then we'll talk through specifics more. So I don't want you to feel overwhelmed at this, is I guess what I'm saying. But the general principles, if we set our traps at 1 foot above mean low or low water, we have to think about what the water level will be relative to our traps across the time that we're sampling. So anytime, of course, that the water is predicted to be higher than our traps, they should be underwater. They're actively fishing. This is also good because cats are not going to climb into a trap that's underwater. When they're exposed, not only are they vulnerable to cats and raccoons, but if we have caught fish at that point and the traps are out of water, our fish are not going to do very well. So we want to make sure our traps are submerged the entire time that they're set, from the time that we put them out to the time that we come back. We also want to capture this overnight high tide, because that's when we're really trying to get as many crabs as we can. So we want to set it at the time when we think they're going to want to come to the trap. So the ideal time to set traps is as the tide is coming back up, because if you wait till here to set your traps, you're going to be swimming. So here, the tide is still low enough that we can get to the depth that we want to put the traps at, but it's almost as soon as we put the traps on the ground, or actually in the water. It's good if you can set them in the standing water. The tide will return and cover it, and then it starts fishing immediately. Again, cats aren't going to swim into our traps. So yeah, I guess this is roughly slack in the middle here. But it also depends on what happens. So you want it after the lower low tide, because that's when the tide is going to be the most likely to get you out to the shore. It'll be low enough to get you out. Then you want it to be followed by an overnight high tide. And then the other thing you have to pay attention to is the second low tide, the higher low tide. And the reason is, go forward just a bit, if you had set your traps here at 2 feet, we would have had this overnight high tide that dropped low enough to send the traps out of water. Again, bad for raccoons and also for the fish that we might have captured during this period. So the ideal time that you return to pick up your traps, if we set them at this elevation, is the very next time that the water drops low enough to expose your traps. And you want to time this obviously so you're not coming back in the middle of the night. And this usually works for this elevation. I also want to point out that we haven't given them to you today, but we will give you in your packets calendars where I've gone through, taken locally relevant tide station data, and interpreted based on these tides when the tide gets low enough to be good for sampling, but also when it's likely to stay submerged the whole time. So I've sort of tried to narrow the guesswork about this, and we'll talk specifics at your site. So arriving just before the tide drops low enough, so this is getting to the point, if you come here, your traps are going to be way underwater and you can't get to them. So you need to wait till the water drops low enough to get back to your traps, and then working to pick them up before the water drops too low that they're exposed. Now, at every site, the soak time will be different. The amount of time that the traps were actually fishing will be different, and that's fine. We can use that to compare between sites as long as we record when the traps were put in and picked up. So we can standardize for that. Yeah, avoid the overnight low tides that expose your traps. So in preparation, we are using frozen mackerel as crab bait. It's great crab bait. It's bloody, it's oily, it's stinky, but it's gross crab bait because it's bloody and oily and stinky. So to sort of mitigate that a little bit, we freeze the bait and we give it to you frozen. You store it in your freezer roughly until you're ready to go out. The thing is, frozen crab bait doesn't attract crabs until it starts thawing and letting that blood and oil seep out into the water. So we're asking you to start by allowing your bait to thaw just a little bit. You don't want it to just be goo by the time you put it in your traps because that's very unpleasant to handle. But just a little bit melty so that the oils and stuff start to act as scent, attracting crabs as soon as you put it in the water. Usually this is just tossing your bait, pulling it out of the freezer like an hour before you plan to be at the site will be sufficient. Maybe it's just hopping in the car, the amount of time it takes to hop in the car and get there, depending on how warm of a day it is. So we have pre-apportioned the bait into these very convenient, tidy portion sizes, where Sean and I went through and measured. You can see the video, I think, on our Facebook page. We have a video of Sean and I cutting and packaging 300 pounds of bait, which is only half of the bait we anticipate using this year. And it only takes you 30 seconds to watch it, although it took us all day to do it. So we have put the bait conveniently in pre-apportioned bags, 1 serving size of bait per bait jar per trap. So you pull out your 1 bag of bait, and these are relatively easy to use bait jars, and you don't even have to touch the bait. You can just sort of dump it in like that and screw the lid shut as I demonstrate how easy it is. I get it stuck. There we go. And then you can just toss it in the trap. You don't have to pin it down or anything because the bait jar is not going to float or get ripped out of the trap. So we'll give you a chance to practice this in just a moment. The traps, the folding traps, don't do it with a laser pointer in your hand. The floating traps, oh, this is set up wrong, that's why. The clips at the top just get clipped shut like this, and we'll practice putting together the minnow traps as well. So 1 bait bag per jar, 1 jar per trap. And then we need to think about how we're arranging the traps on the shore. We're setting a string of traps. And we want them to be situated in a way that will make it roughly even how attracted the crabs are to each of the traps. So we want to put the traps in a line parallel to shore at about the same elevation. So they should be in about the same depth of water when you set them. Alternate the type of trap, minnow, fakui, and setting them 10 meters apart. So your site looks a little bit like this, and let me walk you through it. This is one thing that I haven't talked about, which is a semi-permanent site marker. And this is a rebar stake, and we'll be capping it with this OSHA-approved safety cap, also for visibility. And so this will help you find your site and work it consistently at the same spot from month to month. This will help you orient where you set your trap string, because you'll always start your trap string just offshore of your site marker. And then you'll always proceed in the same direction down the shore with your trap string. Now, the distance between your site marker and your trap in terms of how far out in the water it is actually not that critical. It'll depend on every site. Mostly we just want you to set your traps in the water, in the standing water as it's returning, or in the standing water. If you're at a lagoon, the site will probably be at the same water level for a while. So then you alternate traps types, minnow, fakui, and they're separated each by about 10 meters. We're not asking you to get out your measuring tape to measure 10 meters. A good rule of thumb is that it's a large stride, especially if you're, like me, kind of short. Or if you're in the mud, you tend to have to take a pretty large stride. Or 2 short steps is another way to do it. You can practice pacing on land. We'll show you another trick when we get to the second part. We actually do have a pre-measured rope that you'll be setting on the shoreline, and you can actually cheat. It's not even cheating at all. And use the rope to tell you where to set the traps. So don't stress too much about the exact 10 meters. That's just roughly about how far we want them to be.
And why do we want them that far apart?
So we don't want the traps to be interfering with each other. We don't want this trap to be stealing crabs from this trap. In addition, when we decided on 10 meters versus 20 meters, there's two things that play into it. One is logistics. Not all sites have enough room for us to do a 100-meter trap transect, so we needed to fit the traps in. The other thing is that green crabs have a very small range, individual green crabs. I was talking about this at the break. Sean has had the pleasant opportunity to glue radio tags to crabs and then snorkel around looking for them with a receiver, looking for crabs in shallow water. He's chased them. We know what Sean does in his spare time for fun. So what he learned from this was that green crabs do not go very far. So Dungeness crabs will travel very far on tides. They've got those very long legs. We talked about helmet crabs also being fast, and they travel very far in and out with the tide. Green crabs do not go as far. They have high sight fidelity, and you'll often find them within, Sean, how many meters?
Within actually 11 about 20, well, actually meters.
Yeah.
10 to 30 meters.
10, interesting number we chose, 10 to 30 meters. So we want to make sure we capture all of the crabs that are nearby, so we're not leaving any gaps, but that we also don't do too much interference between the traps and have low trap catches just because this crab got sucked into that trap. So setting the traps, actually setting up and deploying them, you'll get some practice putting traps together. There's a little bit of a learning curve, especially with the minnow traps, and a little bit of force that sometimes has to be exerted. But you clip the traps together, and then we've given you stakes to stake the traps down. These aren't weighted because that makes them very cumbersome and heavy to carry. But essentially what you want to do is you want to have your trap and drive the stake all the way through, like you're killing a vampire, I guess, all the way through the heart of the trap, and there's a little hook on the end that pins it down. There will be some water movement with tidal exchange, and so we don't want to lose these traps for a number of reasons. We certainly don't want to participate in any ghost fishing. So pinning it down so that it goes all the way through the trap and really secures it to the bottom. And in the same way here, this is going to fall apart when I pick it up. Just one stake, yeah. And same thing all the way through and pinning it down. Sometimes you have to move the stake around a little bit to find a spot that will let you go all the way down. Doing it at an angle, if you're hitting stuff that's fairly shallow, you can do this at an angle and it'll still be pretty secure. Yeah, that's a great question. Yeah, so these orange tags say WSG for Washington Sea Grant Research, and then give the phone number that will harass Eileen Herman at the front desk. We haven't gotten any questions yet. So the traps are all tagged, and in fact, so you can imagine setting this minnow trap. You can sort of orient it in any direction. So we'll ask you to set it with the trap tag on the top, so that nobody has to rip up your trap to figure out who to call. The site marker will also have a tag on it, and that's the thing that stays there when we leave the site. So we really want people to be able to know that it's not anything that's not supposed to be there. We haven't yet. It hasn't happened at all to us yet, but we pay attention to that at sites, especially when we're working at a new site, talking to people about what they know about how people use the area, particularly younger people, not to stereotype, but yeah, or people who, if they don't want our traps there, we again try to maintain good relationships. This does have some threatening language that I'm sure is not at all enforceable about punishable by law, fine, and/or imprisonment may result. So, if anyone has the patience to read something like that, maybe that'll stop some of them. I hope. So that's the easy day. That's day one. Again, it shouldn't actually take you much more than. I would be surprised if it took you a half an hour, especially the second or third time you do this. You'll be pros. Coming back the next day, so you set your traps for the overnight soak. Come back the next day as the tide is ebbing, and you're going to be counting and identifying all of the organisms in the trap. You'll get the opportunity to walk through doing this, but roughly the steps are that we ask you to take the trap and empty all of the contents into one of these 2 sorting bins. We're giving you 2 bins, one of which has this scale bar on it, and one of which is blank, just because I didn't need to do scale bars on every bin, so I didn't. And that also helps you keep track of the purpose of the bin. So you get your trap, you can take out the bait, or you can just dump it into the bin, either way. And dumping, sometimes you have to encourage the crabs a little bit, especially the ones that are in these corners, that their claws are good for something, and sometimes it's holding on to the mesh in this netting. So it's, get all of your animals into this bin. Put a little bit of water into this bin. So if we have fish in here, we obviously don't want them to be out of water while we're collecting information on them. So put a little bit of water in this bin. And now that you have all of your trap catch in that bin, we would like you to take a photograph of the entire trap catch. This is essentially a backup data source for us. And in order for it to provide all of the information that we would need, we've created these photo cards, which have your site number, site name, and the date that you collected the information on. And they're individually numbered by trap, so we have ones for both minnow and fukui, trap 1, 2, 3, 1, 2, 3. In terms of the order of your traps, to me, it doesn't matter whether it goes 1, 2, 3, 3, 2, 1, 2, 1, 3, et cetera, as long as the number here matches up with what's on your data sheet for that trap. So you're going to take a photo. Almost always your phone is good enough to take a photo. You can take one with your phone, or you can use a digital camera. Either is fine with us. Mostly, and I want to point out here that we are aware. So we have had sites, I think our record is something like 513 crabs in a single trap. So you can imagine that in something like this, it's 4 or 5 crabs deep. So you're not going to get an individual picture of each crab, and that's okay. At that point, that's 98% Hemigrapsis oregonensis. And what that gives us the ability to do is A, see and identify any fish that were in there. We have not gotten a trap of 500 fish. So we usually get, this is not uncommon for the number of fish. These are tide pool sculpins in this case, where you can see what Sean was pointing out, that they sort of curl up sideways some of the time. This one here too. But this gives us an opportunity to spot check. So if you go out 1 month and we get a data sheet that says 500 Hemigrapsis oregonensis, 4 Hemigrapsis nudus, and the next month it says 500 Hemigrapsis nudus, 4 Hemigrapsis oregonensis, that might sort of be a surprising thing, but we can take a quick look at your photo and see, was that really all nudus the second month, or was there confusion about identification? So that's why we're asking for this information. And that's why we're asking that if your trap is empty, and therefore your bin is empty, that you actually take a picture of an empty bin. It sounds silly, but these zeros are very important data for us. So making sure that a 0 is really a 0, and that it wasn't that the trap got swept away, or we lost the picture, or something like that, we want to know that we have complete data. It can be a little tricky to get good photos. You have to be aware of the sun and the glare, and sometimes having somebody on your team be a shadow maker to avoid any reflections. Sometimes take a few photos and then look at them at home to see which is the best one, because you'll be sending us one. So we want to record the species present in the trap before we set them free. So the photo is really a backup. We need you as the closest, most immediate contact to those things will have the best perspective on actually identifying them. For all organisms, we want the species name and we want the total number. So how many hairy hermit crabs are there total? For crabs, and there's an asterisk there for a reason I'll tell you in a second, for crabs, we want the size information only for the first 10 males and females. So if you get 500 crabs, which is pretty rare, but if you get 500 crabs, we do not want you to measure all 500 crabs across 6 traps. That's just, it's more information than anybody needs. And I'll walk through I'll unpack this a little bit in a second. Crabs has an asterisk because you don't have to do this for hermit crabs. As Sean mentioned, the grainy-handed hermit crabs retract into their shell, so identifying sex and size, the measurement is different. It's too complicated. You don't have to worry about that. But for all of the other species of crab, we would like to have the size of the first 10 males and 10 females. Okay, so males and females. Does anybody know how to tell a male crab apart from a female crab? This was, I know, on your pre-test. Go for it, Bob. Or do you mind sharing your experience?
Sure. The abdomen, which is folded underneath like a shrimp abdomen curved around that made itself into a crab,
Exactly.
is relatively wider and triangular in a female than it is male. They're both triangular, but the male has narrower.
Perfect. Yeah, perfect. And
All relative, so you have to see a few.
yes, exactly. This is exactly true. So this is a picture of the underside of a crab. There would be legs coming out on either side. It's a little abstracted. But the female, the abdomen here is much rounder. It takes up all of the space next to the legs. The male is long and narrow, and there's a bit of the underside of the crab visible underneath to either side of the abdomen. And now, it does vary from species to species, and this is Hemigrapsis organensis, which looks almost exactly like this diagram, very conveniently. But in some species, it's a little harder to tell. This is rock crabs. These are red rock crabs. Is the top the male, or is the top one male or female? And bottom one, male or female? That's right. This is the female rock crab. The difference here is that you can still, even though this is very narrow compared to this, for the male, you can still see some of the underside of the crab, the plates, visible between the edge of the abdomen and the legs. And here, that's not the case. The edge of the abdomen runs right up against the leg joints. So that's how you tell male and female crabs apart. Nicely conserved throughout almost all the crabs you'll see. Now the measurement, how we want to record size, is similar to what you do if you crab recreationally. What we're going for is the width of the carapace, the back shell. And the difference here is that if you're familiar with recreational crabbing, you set your gauge just in front of that wide point. As Sean mentioned, it's a really convenient notch for setting your gauge. Unfortunately, we're crab biologists, so we want you to get the width of the entire shell, including that last wide marginal tooth. And so on Dungeness crabs, you'll set it to the edge of the teeth. For hairy helmet crabs, it'll be this from edge to edge of the widest point. So on rock crabs, we talked about the last two marginal teeth being kind of almost similar in width. It's not as important, but whichever one, if there's one that's slightly wider, if they're the same, don't worry about it. One is slightly wider, go for that one. Back shell at the widest point. We are taking carapace width in metric in millimeters. And we'll walk through how to do that. You guys should all have calipers in front of you. Who here has used vernier calipers in the last year? Great. Okay, so we have a few experts that can also help troubleshoot, and we're going to walk through it right now. So vernier calipers have two ways that they can measure things. They can use these short posts on top to measure the inside diameter. If I wanted to measure how wide this was, I would set the posts inside, stretch it all the way out, and then read the scale. That's not the measurement we're taking. So we could chop those off, but we'll just ignore them instead. The bottom posts measure the distance between the inside of the two posts. So if I wanted to measure the outside diameter, I would set it like that. So what I'm gonna walk through this linearly, and we'll talk about how to read the measurement as well. Sean has taken my crabs. That's okay. So in order to, if this is a crab, we're going to imagine this is a crab. The order in which I would recommend you do this is to set the gauge on the crab, then carefully lift the calipers away without changing the distance, and then put the crab down before bringing the calipers up to your face to read the tiny numbers. So ideally, you would not bring a crab up here. You can imagine. Okay. We've all been down these roads. We tried. It's all trial and error. Now, when you put the crab down, this is why we've given you two bins. You don't want to put the crab back into the bin where you just pulled it from because, there's 400 crabs in there. Which one did I measure? If you put it on the beach, it will undoubtedly walk away. And if you've missed your measurement or forgot to look at what sex it was, you've lost data at that point. So the best thing to do is to put it into the other bin. So you're sorting crabs one at a time from one bin into the other so that you can keep track of crabs. You can release them all at the same time in the right place. So you don't want to just dump them up on the beach where you're working, because they're going to have to walk down to the water, and there's a lot of hungry seagulls that are going to be following you, looking to pick off some crabs. So sorting them from one bin into the other. Now you have your gauged, I just measured a crab, it's this big. You have your gauged calipers, and we've put the crab down so our nose is safe. The way you read these vernier calipers, we're only interested in what's on the bottom of the calipers. You'll notice that on these white calipers, this is actually in inches at the top. So that's part of why we're really not interested in that scale at all. We're only interested in metric. You'll notice if you close your calipers all the way, this 0 line will be continuous with this 0 line. That means the distance between the two posts is 0 millimeters. So this 0 line is what we're using to tell how far apart the posts are. And we read it with which of the lines on this top scale it's most closely lined up with. So in this case, this is in centimeters, remembering that 1 centimeter is 10 millimeters. So we have 0, 10 millimeters, 20 millimeters, and then each line is 1, 21, 22, and this line is close to between 23 and 24 millimeters. Now, there is a way to extract information to another decimal point using this, but we're not asking you to do that. We're just asking you to go to the nearest millimeter. So which of these two lines this is closest to? I would say this one is closer to 24, so I would round this to 24 millimeters for this measurement. So in this case, if you think it's exactly in the middle, round up. So if it were 23.5, go ahead and round up to 24. So what I'd like to do now is just take a practice. If you guys could all pull out your ID guide, we're going to measure us a green crab. I'd like you to measure the carapace width of the line drawing of the green crab at the top in the middle of the ID guide page. And Kate and Sean and I can help walk around and help you if you're having trouble remembering how to read this. The line drawing, that's right. I think it's the perspective on the other one's kind of weird. Yeah. So the line that the 0 is next to, because you'll notice that if you close it all of the way,
Yes.
that 0
It
line
lines up.
should be lined up to that 0 line.
Okay, so I'm measuring it by, I'm confused.
Sure.
So I'm looking at this, 10, 20, am I using the 0 line? The
Exactly,
0? Okay,
yeah.
so right there, 2, 1, 2, 3.
Mm-hmm.
Okay,
So 23 total.
23.
Is that?
Yeah.
Yeah.
Drawings.
And the magnification on the printing might be a little different too. Long or wide? So did everybody get 78 millimeters? Is that what everyone got? Oh, I guess I've been doing it wrong all of this time. What was the measurement that everyone got? Or if you want to shout out what you got. Yeah, so we get repeatedly, when we do this ourselves, we get about 22 to 24. The precision on these fine instruments that is about within 1 millimeter. So 22 to 24 is an acceptable range. If you are getting 78 millimeters, we'll discuss further.
That's one of the reasons that we don't care about the point anything else, right? That's not going to be meaningful. We're
Right.
not that accurate.
Yeah. Yes. Okay, and you'll get chances to do this also. We'll practice this in just a few minutes. We'll get people out. So this is the data sheet. It would be great if you guys could take this out. It's in your packet towards the back. This is the data sheet that you're using for trapping. And we're gonna fill it out together here, and then you'll fill it out when you're practicing with each in your breakout groups. So the metadata at the top is just what dates are you trapping, your site number, which we'll give you, and your site name. This part is your volunteer timesheet. This seems like a lot of information for us to be collecting on you, but what this has to do with is essentially the workers' comp that covers you as part of participating in this program. In the unfortunate event, if you were to be injured, we need to know what actual times you are working start and finish on the 2 days. This includes travel time, any other preparation or cleanup. So anytime you are contributing to the project is considered in this timesheet. Obviously, you should probably use names so I can actually tell who you were, but this is our we all have names, code names for ourselves, I guess, nicknames. Over here is where we record when the traps went in and when they come out. You set the date and time, and the time, since it's going to take you a little bit of time to get through setting all of the traps, you want the time that the last trap went into the water. So this is sort of going to be a conservative estimate of how much time the traps were fishing, but really you'll start to see crabs wander into your trap if there's a lot of crabs at your site. Even as you're sort of within 20 minutes of setting the traps, they start to be attracted pretty quickly. Same thing when you come back the next day, only we want you to record the time that the first trap was retrieved. The traps are not really going to be fishing once more, especially once you're on site and you're sort of tromping around near them. The crabs are going to be less likely to go into the traps. So again, this is a minimum soak time, essentially an estimate of a minimum soak time for the traps. And so that everybody's doing it consistently, last trap set, first trap retrieved. Record a dominant weather condition. We are certainly familiar with how frequently and rapidly weather can change here, but what do you think the traps predominantly experienced over the last 20 hours of trap soak or whatever? Was it predominantly cloudy or was it totally clear? How would you describe the weather to a friend? Don't stress too much about this. Okay, so now here is the meat and potatoes. You can see we've dedicated most of the real estate on the front and then all of the real estate on the back to recording the actual organisms in the traps. Each wide row gets one species per trap. So each species in each trap will get a whole row dedicated to it. And we're going to walk through filling this out, and I'll show you a couple of contingencies as well. So the first thing you record is what type of trap is it? Minnow or Fukui, and what number of the trap is it? And this is the area where it should match up with whatever you've taken a photo with on the photo card. So this is minnow trap 1. This would be, if it were this card, it would be minnow trap 2. This is where you record the species code. Rather than writing Pagurus hirsutisculus 6 times, we'd rather you just write 4 letters that will help you identify it. There's a cheat sheet both in your ID guide, and then each of the binders, clipboards will have this cheat sheet stuck to it that gives you the 4-letter codes as well and a few extra tips. So you write in, we had Metacarcinus magister here. Now you record the first 10 males and 10 females sizes. So the first size for 10 females will be in these 10 boxes, and for males will be in these 10 boxes. So here we only had 4 Metacarcinus magister in our entire trap, so we measured all of them. And they were this 67, 75, and 60 millimeters, and one female that was 55. Then we also totaled them, sorted by males and females, at the end of the row. This is a space that you can use to tally. If you do have a lot of crabs, it's easiest to just have people read off male, female, and have somebody keep a running tally. So this is the space that you can use for that. You might need more space than one row allows, so just feel free to continue on the next row. You should have plenty of space between the front and the back of a data sheet to collect all of that information and use up a few extra rows. I don't think even last year, based on the number of species in the traps, that we even went onto the back of one of the data sheets. So there tends to be a relatively small number of species within a single trap. Now, if you're in a situation where you have more than 10 males or 10 females, so here, this is the same trap, but now we're measuring HEOR. So we do a new row for a new species, minnow 1, Hemigrapsis oregonensis, hairy shore crab. And in this trap, we had a total of 15 males and 7 females. And so we measure all of the females because we need up to 10. But now we only have to measure 10 out of the 15 total males. How do we choose which males to measure? Well, yeah, the fancy ones. Well, the quick answer is we actually try not to choose them. Ideally, in this situation, if we want to capture a snapshot of what the population really looks like, we would assign random numbers to these. That's actually impossible when you don't know how many crabs you're going to catch, because we have to know what the probability of any individual crab would be. We don't know that. So what we do is an ecological type of sampling that is, it's official ecology, I promise you, but the word for it is haphazard sampling. And it sounds sloppy, but it's actually a very thoughtful approach. So the idea of haphazard sampling is to get it as close to random as possible, meaning that every crab in that bin has the equal chance of being sampled, regardless of its size or how pretty it is. So what we're trying to do is reduce human bias and reduce crab bias. So human bias, we might pick the pretty ones or the small ones with the tiny claws that don't look like they're gonna pinch us very hard. The crabs also tend to segregate themselves by size. So one idea would be to work from one side of the bin to the other, but then you get all the big crabs one side. So yes, the approach is to look away slightly and gently mix the crabs, and then pick the first 10 that come to you pick the crabs that come to you until you get up to 10 males and 10 females. Then, since you're not measuring them after that, you just get through all of the crabs, sorting them male, female, tallying them up, males and females. If you collect a bin full of red rock crabs that are this size, I do not recommend this approach. Please don't do that. If you get a trap full of that many large red rock crabs, we probably have your traps in the wrong spot. So if you get any one rock crab that large, we probably have your traps in the wrong spot. So that's a situation that you won't face regularly. Okay, so the haphazard sampling sounds sloppy, actually very important. After you have measured all of the organisms in one trap, they should all be sorted into your sorting bin, and you can go and set those free before you start on your next trap. So you want to walk out, put them back in the water, find an area that's not just covered with gulls waiting to pick off the crabs that you just measured as they're all disoriented, and let them go, and then you can go grab your next trap. So next trap, Fukui 1, and here I've asked you to take up a row even if the trap is empty. This is another way we record zeros. Same way if we take a picture, here we know that Fukui 1 was empty. You don't have to enter anything else in the row. Now, we moved on to the next trap, Minnow 2. LEAR is our staghorn sculpin, Leptococcus armatus. You do not have to measure or record the sex of fish. So here you just write a total number for the species. The same would be true of hermit crabs as well. Do it in the same way. What we want you to do is actually, when you get to a trap, count and release the fish before you move on to the crabs. So what you can do is you have a little bit of water here, a little bit of water here to keep things happy. 1, 2, 3, 4 staghorn sculpins, and you can sort of scoop them. We'll give you a pair of gloves to avoid the pokers, the pokey bits. Scoop them into here and have somebody run them down back to the water while these started on the crabs to get the fish back as quickly as possible. Some of, especially the shiner perch, are a little more sensitive. We want to get those guys back in the water very quickly. Count and release the fish first, then move on to the crabs. Once you get through all the crabs, you can release all of those at once, and you're good to go. Okay, if you find a green crab, we put Jeff's picture here, and he's not here today. Jeff Adams is the one that we want you to contact first. Jeff is highly available via his cell phone and can receive texts with pictures if you have the capability. Any time of day or night. We strongly encourage you. It's urgent. Green crab matters. It's very important. So,
You need a friend to talk to?
yeah. Babysitter? I don't know. What else can we volunteer him for? Water your plants while you're on vacation? Yeah, right. So this is his phone number. Go ahead and write it down on your paperwork. We can email it out to you certainly, but we're not going to put it on the website to protect some of Jeff's privacy, at least. But he loves taking pictures. He loves taking questions and identifying green crabs. So he will get you on the path to what to do next. The upshot is you're going to keep the crab, not put it back in the water. Stick it in one of the Ziploc bags that your bait came in. Those can be handy for that. And the most humane way to kill a crab, in contrast to what those people in that video were doing, is to stick it in the freezer. So stick it in the freezer, keeps it from rotting as well, and we'll verify the identification and come get it and take it out of your freezer. So we're not asking you to store dead crabs in your freezer indefinitely.
Can we assure you that Jeff has this task whether or not he attends the training?
Yes.
That training means Jeff is there.
We throw him under the bus.
It's not only because he's not here today.
Yeah, that's what he gets. That's what. Yeah, it's the. One of us will.
Do we need to call him while we're standing in the mud?
If you can, that's the best thing. If he might take a little while to get back to you. If you can send him pictures by text, that's great. If you should be able to take pictures. But if you're using a digital camera to do that, then you might need to wait to get back to email pictures. But he'll probably ask you identification questions just to double check that that's what it is, and then that'll start the chain of following up with you. If you can't identify an organism because it's something that's not on the guide, or there's a little bit of, you can't tell if it belongs to one or the other, we would like to identify it for you. We are a one-stop identification shop. And in the meantime, before we can get the information back to you, you can use a placeholder in that species name area. You can release the organism unless you think it's a green crab, and then we want you to take a bunch of pictures just of the individual you can't identify from different angles so that we can see more features, and that makes it easier for us to be able to tell what it is. I put this here because I'd like you to include some indication of scale. This is a beautiful picture of a snail, but it is missing a key piece of information, which is that this snail is very tiny. So if you have something in there that you can stick for scale, your calipers, you have calipers there, even your data sheet or your photo card, those are things we know what size they are, so we can get an idea of how big it is based on that. Cleaning and storing gear, again, not super glamorous, but important to the longevity of your gear and the project. Any large debris, like algae sometimes will sit on top of this, float in and sit on top of this, or eelgrass, you can just leave that at the site. Try not to take things with you. If your traps get really muddy, rinse them in, just sort of wash them off in standing water that's at the site. So like if there's just a little tide pool or whatever, just rinse off the mud there. Take your bait home with you to discard. I am sorry to send you home with this fish that's been frozen, then unfrozen, and then sitting in seawater for 24 hours. And we talked about this, we don't want to discard a lot of bait at a site where, especially low energy sites, it's not going to get washed off shore. We could be attracting animals that aren't necessarily there. Somebody else comes down the beach and thinks there's been a major fish kill. It's just kind of gross to have rotting fish there. So take it home, compost it, plant it in your garden, feed it to, maybe you have opossums at home that like that sort of thing. I don't know if you really want that, but right, you will have opossums if you feed them. When you get home, it's important to rinse the traps, and ideally all of the gear, but especially the minnow traps and the stakes with fresh water. They are galvanized steel, but they will rust because these aren't the fanciest traps that you can buy. So this will help us increase the life of these traps and make them easier to deal with. Sometimes the mesh on these Fukui traps it's a flexible plastic, so sometimes it does rip. A lot of the times you can actually repair that by sort of stitching it together with small zip ties. You can sort of knit. It's kind of a fun arts and crafts time. So we'll give you some zip ties, and you can repair those at your leisure. And however, if the trap gets totally plowed by a boat or I don't even know what, let us know and we can replace any gear, or if it gets lost as well. These traps come from Japan. We import them from Japan. They're not actually that easy to come by. These are very accessible at most outdoor sports stores.
I have a question about the laser trap.
Uh-huh.
I would have to go home to do that, right?
Right. But sometimes, some places like Bayview State Park actually has a place, a freshwater place, and so it's best to do as much cleaning as possible on site, not only for the sake of your car, but also so that you can avoid moving species to a new place accidentally. And storing traps in a dry covered place so they're out of direct sunlight. This plastic is probably going to degrade with UV over time, and also so you don't have small creatures nesting in them when they're not sitting in pools rotting. So I think, and then one last step. I always almost forget this. Submitting your data. So because you're going to be collecting data over a long time scale, and because we'd like to stay abreast of what's happening in the field, we're asking you to email an image of your photo of your data sheet after it's been completed. So you can actually typically take this, you could scan it if you have a scanner. That makes the nicest, I'll tell you, that makes it the easiest for me. But you can also almost always take good enough pictures with your phone. Our students do this all the time. They take pictures of test keys so that they can go back to their own test and read it, or instruction sheets, things like that. We've asked you to rename the files based on this convention. It doesn't matter what type of image file you send us, unless it's like, some proprietary thing, but any basic one that your phone will take will work just fine for us. And then if you have to go onto the back of the sheet, it gets a number 2, but most of you it'll just be 0.1. We're also asking you to email us each of those photos that you took of the traps. Of course, it would have a picture of the little photo ID card in it, and these are named in a different way. So this information is all included in your handbook. So you'll be emailing us for trapping at least 7 files each month, again, even if they're empty there. So, and then at the end of the season, we'll ask you to send us as a batch, instead of doing it every month, the original data sheets. You'll have a pile of them. We'll give you an envelope that has our address on it, and you plunk it in the mail, and it'll find us, and it'll go into the record for posterity. All right, I'm sure there are a lot of questions, and if there are any overarching ones, I can ask them now, or you can ask them now, or we're going to just quickly go through what it takes to set up a trap and then fill out the data sheet, come back the next day and fill out the data sheet. We're going to divide up into 3 groups. Kate, I, and Sean will each lead one. So feel free to divide yourself up. Yeah, Sean. Yes. So please bring with you, we'll bring all the gear. In terms of paperwork, we'd like you to bring your handbook. We'd like you to bring this data sheet that we just talked about, the trapping data sheet. And there's a special identification guide in the very back of your packet that has some fauna that are unique to the ecosystem of this classroom. So you might not have encountered these before, and that's fine. Is that everything, Sean? I think we don't need the ID. And your calipers, because we will be measuring. And a pen and a clipboard. Okay, so Sean, where are we headed?
So, you just want to stay where you're at?
Sure, I'll stay where I'm at.